In this step 5 video, we're going to look at how we can take the output from step 2, LD score regression, and the output from step 4, the aligned summary statistics across the included GWAS, to now include the effects of individual genetic variants, i.e. SNPs, within the model using the userGWAS function. userGWAS takes 3 required arguments. As I just mentioned, the 2 main pieces are the output from LD score regression and the output from that SumStats function that we just ran in the previous video. And then the third argument, just like with userModel, is the model syntax itself, where you're going to include the effect of a SNP using the general notation of SNP, which we'll see in just a second in some example model syntax. Behind the scenes, what userGWAS is doing is it's taking the output from LD score regression and the output from SumStats for each SNP. So this is just showing the covariance between a given SNP and schizophrenia, bipolar, and MDD. And so it's taking this output from LD score regression, which has the genome-wide genetic overlap across the included traits, and then the output from SumStats, which reflects the covariance between the SNPs and the included traits, where part of what SumStats is doing is it's rescaling the output so that it's on the same scale as the output from LD score regression, so that when you run userGWAS, the 2 pieces of output can be appended to one another, so that what userGWAS is doing behind the scene is it's actually recycling out this first column here for each SNP and appending it to the matrix, and then using that as input to the model in the same way that you were doing for userModel in the 3rd step in the prior video. Each time the model is run, it's recycling out this covariance between the SNP and each of the traits, and then this part with the LD score regression output is staying the same. I wanted to also highlight some frequently used optional arguments. One of them is the sub argument. I highly recommend specifying this argument. What this is doing is it's saying that you want a specific piece or pieces of the model output saved for each SNP. So for each SNP, it is rerunning the model, estimating the effect of the SNP within the broader model that you're specifying. But you probably don't want every piece of that model output every time. For example, we're going to look at a model in which the SNP is predicting that internalizing factor, but I'm not particularly interested in the factor loadings for each individual SNP because those are more or less going to be the same, nor the residual variances of those 4 psychiatric indicators. So in our example, we're going to look at setting sub to only save the effect of the SNP on the factor. If I don't do that, the size of my output files is going to be at least 10 times larger because it's saving every other row of that model output for each SNP. The second optional argument is std.lv, so we actually saw that with userModel. This again stands for standardized latent variable, and it's whether or not you want model identification to use setting the variance of the latent variable to 1. Parallel is an optional argument that you'll often use in practice. So this reflects whether or not the userGWAS function should submit jobs should submit. Parallel is another argument that you're often going to use in practice. What this does is it says whether or not the computer should estimate the model across the different cores on the available system. And if you are setting parallel to true, which I typically am when I run a GWAS in GenomicsM, I'm also have the option to say how many cores I wanted to use. The default is it's going to use 1 less than is available in the environment that it's automatically detecting. I would note that each run is independent of the other, and that I also am typically running a multivariate GWAS on a computing cluster or on a cloud system, not on a personal laptop. Everything up to this point could easily be run on a laptop. It is this step that just if you were running a univariate GWAS using raw genotype data, here I would also recommend running on a computing cluster. If you don't have one that's available to you and you have a relatively simple model like we're looking at in this example here, it would be possible to run it on a laptop, though it's going to take at least a few hours. But for more complicated models, say with 10 traits, multiple factors, it's not going to be pragmatically feasible to run it locally on your compute environment, and you're going to want to get set up on a cluster so that you can not only run in parallel, but potentially submit multiple jobs that each themselves are running in parallel. And a note related to that point of submitting multiple jobs is that each run of userGWAS is independent of the other. So you might have millions of SNPs from some stats. You can split that any way you want across jobs. It's not going to change the output for each individual SNP. Okay, so finally, a newer argument available is QSNP, which is whether or not the function should automatically calculate this QSNP heterogeneity metric for each latent factor. So just going over again what that is. So QSNP is effectively comparing the fit of a common pathways model is being shown on the left here to the fit of an independent pathways model that is being shown on the right. And so the question is, a single pathway from the SNP to the latent factor sufficient for describing the pattern of relationships between the SNP and all 5 indicators that are being used to define this particular latent factor? So you can think of QSNP both as a QC metric and a result. As a QC metric, you would want to prune for the factor estimates, the SNP effects on the factor. You would want to prune out anything that is genome-wide significant for QSNP at a minimum, and likely the entire set of SNPs that are in LD with that genome-wide significant QSNP, because this reflects a gene region that does not operate via the factor, as indicated by the fact that a single path from the SNP to the factor does not adequately describe the relationship with the individual traits. In other instances, you might have traits where they are so highly correlated that you're specifically curious to know what SNPs may deviate from that model. So let's say you have 3 traits where there's a question about whether or not these actually reflect distinct phenotypes, and indeed the genetic correlation is exceedingly high, maybe in the 0.8 region. So you have specific research questions about what are the SNPs that actually may distinguish these in ways that the SNPs that are significant for QC would be pruned from the factor, but then might be examined in their own right as a result, highlighting those distinct or divergent pathways across these overlapping disorders. If you have multiple factors, you can get a QSNP specific metric for each factor. And when you set QSNP to true, it's automatically going to calculate that metric for each. It used to be the case that you had to run a separate independent pathways model and do the model comparison relative to the common factor model in the way that I was showing in those common and independent pathways models. Now I merely show that to be illustrative of what QSNP is statistically indexing, but there is now a closed form solution that userGWAS employs so that you do not need to run separate models to get QSNP, and it's going to provide a separate metric for each factor. I showed this particular factor model as one way to illustrate how you can think about factor-specific effects for QSNP. So in this 5-factor model of 14 psychiatric disorders, we find a separate set of QSNP effects for F5, where we find that genes involved in drug metabolizing pathways for these different substance use disorders are often significant for QSNP. So for example, you get genes in the nicotine receptor pathways that are significant for this substance use disorders factor, which makes sense. The nicotine pathways are not capturing the shared risk pathways across substance use disorders. They are specific to nicotine. And so the fact that it's showing up as QSNP significant is really just kind of illustration that this metric is working as intended. So again, you used to have to estimate these separate models, and lots of credit to my previous postdoc, now lecturer at Queen Mary University, Dr. Izzy Foote, who introduced this closed form solution in a recent Nature Genetics paper. And the QSNP argument is and has been live up on the main genomics SEM branch for about a year now. Okay, so here we're going to start by loading in our 3 necessary arguments. So I'm going to load in that LDSC object we created in the second step. Then I'm going to read in that SEMstats file that we created in the last step. Again, just for the purposes of this video, we've subset that to 1,000 SNPs from chromosome 4. But in practice, you're going to have millions of SNPs that can be split up across jobs in a computing cluster in any way that makes sense for your computing environment. And then finally, the third necessary argument is the model syntax, where this first line here mirrors what we had for user model. It's just the latent internalizing factor that we're calling F1, defined by these 4 disorders. And then the next line is the effect of the SNP on the factor, the multivariate GWAS part that we're implementing with this userGWAS function. And just as a note of caution, it's actually pretty common that a mistake would be that this part is left off. totally makes sense. You're copying over code from user model and you just forget to add on this part. So just remember that you do need to specify this. For the optional arguments here, I'm just interested in the effect of the snip on the latent factor. So we're setting sub here to specify only saving that parameter. This can be a vector if you have multiple parameters of interest. So if we had a multi-factor model with 2 factors, we might include something like that, where we want userGWAS to save 2 parameters, including that F2 effect. But here we're just looking at that 1 parameter. Just for the purposes of the video, we aren't using parallel because we're only looking at 1,000 snips, but in practice, pragmatically, if you want to get results faster, you're going to be setting that to true, and again, ideally running on a cluster. And then finally, we're setting QSnip to true to get that heterogeneity metric for each snip for that factor. So then we put all those pieces together to run the userGWAS. So one thing I want to note here is this fixed measurement part, where a few years ago we introduced this argument that is default to true, which says that the measurement model, which is to say the factor loadings, should be fixed across SNPs. And this is done for 2 reasons. 1, it speeds up run times considerably. And 2, it also improves interpretability across SNPs because now you've got the same model that is being applied across every genetic variant. Whereas when you are freely estimating that part of the model, it could deviate in ways that are frankly hard to suss out because it's not easy to look at millions of different pieces of model output across all of your SNPs. Okay, so now that's run, we're going to take a look at the first 5 rows of output just to make sure everything looks sensible here. Here, userGWAS will save things in list objects. We only had 1 argument for sub, and so there's only going to be 1 list object here. But if we had that second factor, it would be saved in the second list object. And then within that list object, we're just looking at the first 5 rows. So here we have some kind of basic information: SNP, chromosome, base pair, minor allele, frequency, effect allele, non-effect allele, the parameter, which is again what we set in sub, which is to save the effect of the SNP on the latent factor, our estimate, standard error, z-statistic, and p-value. Some model fit statistics that frankly are probably not super relevant because they were mostly used previously to calculate QSNP using those model comparisons with follow-up models. But again, that's no longer necessary. When you snap QSNP to true, it's using that closed form solution so that now we are automatically getting that QSNP metric. So I would pay more attention to these last columns here, where this is going to give you the SNP-specific heterogeneity metric, where more significant p-values here indicate that a SNP more strongly deviates from the factor model. Here, what I want to do is look at whether or not we have any genome-wide significant hits, less than 5 times 10 to the negative 8. This is just a random subset of 1,000 variants on chromosome 4, so unsurprisingly we don't. Similarly, we don't have anything that is QSNP significant, but these might be the kinds of things that you start to look at to make sure things are looking sensible, that you have some hits, but it's not the entire genome that is showing up as genome-wide significant. From there, you can do the standard follow-up that you would do for a GWAS, creating a QQ plot, looking at a Manhattan plot. The sort of unique aspects would be doing quality control based on Q. But having done that, you could also look at things like polygenic score prediction for these latent factors and doing standard biological annotation pipelines, in addition to the within genomics and biological annotation pipelines, like using gene expression and functional annotation that we're not going over in these videos, but you can see the GitHub for more details on those more recent additions.